My name is Anna. I'm going to be the technical moderator of the session. I'm very, very happy to welcome all of you here in the special session on biological fouling during the Marine Research and Education Conference. In fact, it is the very final session of the conference. Thank you for joining us today. And with that, we are going to start our session, and I am going to pass the microphone to the scientific moderator of the session, Alexandra Chava from the Institute of Oceanology in Moscow.
Hi all. I'm very glad to see you, and I hope we will have fruitful discussion after the presentations. And I would like to start with inviting Professor Vadim Makievsky from Institute of Oceanology in Moscow with welcoming presentation, actually one welcoming slide about our session, please.
Thank you, Sasha. I am very glad to say hello to everybody and to welcome all of you to our session. And I just like to highlight several points concerning the biofouling, which is a really well-known process in general, but there are at least two topics which are much less known. The first is what happens below the depths of several dozen meters. The main driver for fouling investigations was shipping and technology, and now with the expansion of technique into the deep, that lead to appearance of many constructions and the development of fouling community in pretty new condition. And it appears that we know nearly nothing about the succession and what happens on the artificial constructions below photic depths, let's say. And there are two drivers of our interest as usual: curiosity, as fouling community are very good model for ecological studies, for understanding the species relations and so on, and also the practical necessity, because fouling communities deep needs our concern, both from technological safety and environmental safety as well, because they might be the stepping stones for invasion species and might drastically change the geographical limits and borders. And at the same time, we know not too much about how fouling communities and communities on hard substrata can alter the nearest soft bottom communities. And this question is open also for shallow water as well. And the second gap in our knowledge, as at least from my point of view as zoologist, is the early stages of succession, the development of biofilms and their role in later succession stages. For us as zoologists or macrobotanists working with seaweeds, it's like a black box. We assume that biofilm can alter the further succession and settlement of larvae, but how it works and what happens in these first hours or days of on the substratum, I hope I learn something about that from our session, and I expect to get a lot of new information, and probably more of the same importance, let's say, is to find colleagues to organize kind of information exchange between people who are interested in the same topics. When organizing this session, we faced with a problem to find specialists interested in deep sea fouling, and it seems that there are still not many of marine biologists who are interested in this matter. And if during this session we find some contacts and start some communication with colleagues, it will be a great benefit of the meeting. Thank you and welcome to our session.
Thank you very much. And so following the theme of biofilms and first stages of succession, I would like to greet Dr. Sergei Dobretsov from Sultan Qaboos University in Oman with a presentation about microbial deep sea biofouling. Please, Sergei.
Good afternoon, everyone. So my name is Dr. Sergei Dobretsov. I'm working at Sultan Qaboos University, and at the same time, I'm an assistant editor of Biofouling Journal. So I hope that we'll see a lot of your interesting publications regarding biofouling submitted to this journal. You can find the link on the first page. And this is the outline of my presentation. So I briefly introduce biofouling and the biofilms. And my talk will be about biofilms, the way how to study them, composition of marine biofilms. I mostly will focus on biofilms developed at the upper layer, although there will be some presentations regarding deep sea biofilms. And then at the end, we will talk about the challenges and the future research directions. Marine biofouling can be divided on two large groups. One is what we actually can see. It's called the macrofouling communities. For example, you can see it on the right-hand side. So this is represented by tunicates, also barnacles, mussels. But for microfouling communities or biofilms, actually you need to have a special tools to see them. And my talk is about these microfouling communities. So biofilms is a form of life of attached microbes. Biofilms is a home for the microbes that are living attached to the substratum and incorporated in the extracellular polymeric substances, or EPS. And these biofilms usually composed of many different species of microorganisms. And biofilms exist everywhere. We can find them when we brush our teeth, on walls, paints, and even in the deep sea. And biofilms has a three-dimensional structure, although you cannot see it from this picture. So this is a scanning electron microscopy picture of a biofilm on the surface of alga. But believe me, they have a three-dimensional structure that we will touch later. So biofilm succession, as it was said, that biofilms are not static, and biofouling is not static, it's developed. And this is then an example of development of the biofilm in Australian waters. And as you can see here, so after first day, there will be formation of the attachment of bacteria to the surfaces. Then bacteria will start proliferating. And then after 8 days, we will have diatoms. And after 16 days, biofilms will be formed. And as you can see here from the picture, there are also mushroom-like structures. So it's a three-dimensional, it's not flat. It's a three-dimensional structure of the biofilms. Again, these 16 days biofilms can be months if we are talking about Arctic or Antarctic waters or deep sea. How to study biofilms? There are several methods, conventional methods that we are using. Most of the time we use light microscopy. This is cheap, but at the same time, this method doesn't allow to visualize microbes, all microbes, because they are most of the time invisible or they are transparent, so we cannot see them. At the same time, we have also scanning electron microscopy, which is a rather expensive method that also required a certain preparation. Usually it's dehydrated and also covered with the gold or other metals at the surface. And of course, it can change the structure of the biofilm, especially this 3D structure of the biofilm. When we want to count microbes, epifluorescent microscopy is one of the standard methods, especially for bacterial counts. And in this case, certain dyes that are stained, DNA is applied, and then bacteria became visible, and we can count them under microscope. Confocal scanning laser microscopy, and you can see this red picture on the right. So this is the example of 3D structure of the biofilm developed on anti-fouling paint. And this actually is a good method that allowed you to visualize thick biofilms. It can be also applied for live biofilms, so there is no need to do any fixation. And the last method, which is widely was used, it is isolation and cultivation. So some bacteria or other microorganisms can be isolated and cultivated in the lab. At the same time, the problem with this method that less than 1% of all bacteria can be cultivated. So we are leaving 99% of all microbes unidentified. But at the same time, we can manipulate with them, investigate their properties, and so on. What are the novel methods that has been developed recently? So the first one is flow cytometry. Flow cytometry usually applied for planktonic cells, not to the attached cells, because if we want to automatically count or sort microbes, we need to actually detach them from the surface. And during this process, some bacteria or some microbes can be damaged. Molecular fingerprinting methods such as DGG, TRFOP, ARISA. So this method allowed us quickly compare microorganisms. They don't allow us to actually identify them, except denaturing gel electrophoresis DGG that in combination with the cloning allowed us to understand who is there. Counting of microbes, it can be done by fluorescent in situ hybridization, or FISH in short. In this case, species level cannot be achieved, but we can look at the certain genus or certain phyla and count and see how many of them are present in the biofilms. Recently, metagenomics methods have been developed. So in the past, cloning was widely used when certain genes are cloned in the, for example, E. coli or some cells that we can cultivate. At the same time, next generation sequencing right now is one of the most common methods to analyze microbial communities. At the same time, it is rather expensive and it requires intensive analysis of the data that are generated, because you will have large varieties of different sequences. And instead of next generation sequencing methods, we have pyrosequencing, which was used in the past, and now Illumina MiSeq sequencing, next generation sequencing is widely used to sequence the whole genomes or the particular genes of microorganisms. Oxford Nanopore, it's third generation next sequencing, which is became now open and bring down the cost of sequencing. And if you want to know more about the methods of investigation of biofouling, I would like to invite you to read our book about biofouling methods. Who is there? If you look at the table presented here, you can see that the top row is actually belong to viruses, but there is a question mark there. So this means that although viruses are dominant in seawaters, this is the data not belong to the biofilms. We have no much information regarding viruses in biofilms. At the same time, the second largest group in biofilms is bacteria, and the third largest is archaea. So bacteria, archaea, and in photosynthetic biofilms, diatoms, these are the most dominant groups in marine biofilms. What are the dominant species of bacteria in biofilms? It depends on what kind of methods you are using. So in the past, culture-dependent techniques were used, and they showed that Pseudomonas and Micrococcus are the most dominant species belonging to gamma proteobacteria group. At the same time, with the employment of molecular techniques, we discovered that there is a high diversity of bacteria in biofilms, even on antifouling coatings, and they are mostly dominated by alpha proteobacteria, although gamma proteobacteria are also present. Diatoms in biofilms, again, they exist only in photic zone, and most dominant ones are Amphora, Navicola, Coconaeus, Synedra, Cylindrotheca. So these are the most common genera that can be found on all kinds of biofilms. Overall, biofilms are widely distributed, so that you can find them at the surface waters, at the deep waters, and there are some difference between sites, seasons, and substrata. If we look at the deep sea biofilms, the number of studies will be quite limited. And mostly these studies are focused on macrofouling communities, so not on microbial biofilms or particular environment like hydrothermal vents. You will see a vast amount of papers published on microbial mats developed in this kind of unique environment. But we don't have clue or much information regarding other biofilms in other parts of the ocean. Mostly it was studied by culture-dependent techniques, so molecular techniques are still lacking, and the studies suggested that dominant species of bacteria, groups of bacteria belonging to gamma, alpha, and beta proteobacteria, as well as we have dominant archaea species belonging to Crinarchaeota. This graph is showing the vertical distribution of microbes in oceanic waters. And what I would like to point out that we have two dominant groups, archaea and bacteria. And in the photic zone or above 1,000 meters, we have a high abundance of bacteria in the surface waters, while if we are going deeper to 1,000 meters and below, we have a dominance of archaea. Again, this is a planktonic microbe, so we don't have much information on the biofilms and their vertical distribution. We try to investigate vertical distribution of microbes in biofilms. So this study was done together with my colleagues in the Mediterranean Sea. And as you can see here, from the right-hand side, we have a substrata attached, panels attached at the depth of 1,500 meters, 2,500 meters, 3,500 meters, and 4,500 meters. So it's well below of 1,000 meters. And this is molecular identified microbial communities. So we look at the presence of a certain phyla in these samples. And as you can see here, so basically we found that all these phyla exist in all these 4 depths. At the same time, their presence was different. For example, if you look at the alpha proteobacteria, 1,500 meters, it was 10%. And if we go deeper, then we have only about 1 or 2% of this group. We also investigate biofilms on autonomous vehicles, in particular buoyancy-driven sea gliders. So this is an important piece of oceanographic equipment. And basically, they have different sensors, and they can be autonomous up to 10 months. And in our case, they were diving from the surface to the bottom and in our case, bottom was about 1,000 meters, and there was about 2 dives per day. So this is the results. We were surprised that we were not able to determine any archaea or macrofouling species developed on these gliders, but we found diverse bacterial communities. And as you can see from this principal component analysis graph, we have basically different communities present in seawater. You can find them with the red triangle. And then we have different communities present on anti-fouling paint, and different communities present on the unprotected parts of the glider. If we look at the eukaryotic microscopic communities, so in this case, differences were not that pronounced, but still we can see the difference between seawater as well as attached microorganisms. So again, seawater microbes or seawater studies are not applicable to the biofouling biofilm studies. Okay, so then we look at the different biofouling on different parts of the gliders. And as you can see, this GT, GB, TT, it's a different abbreviation for different parts of the gliders. And if you look, for example, on W1, this is a wing of the glider. You can see that it's dominated by green, which is gamma proteobacteria. But if we look at the other parts, gamma proteobacteria are not really a dominant group of microbes. At the same time, we also see the difference between the top and the bottom of the glider. So for example, if you look at the GT and GB, you can see that this is a glider body, and T stands for top, B stands for bottom, and you can see that bottom has a large amount of beta proteobacteria, which doesn't exist a lot at the top of the glider. So different communities on different parts of gliders were different, and this probably was because of the orientation and different flow. This is a slide from, not slide, but picture from the recent publication in BMC Microbiology. Unfortunately, these investigators, they use a culture-dependent technique, so they isolate microbes from water column from different oceans. And you can see that they have sometimes 100, 150 isolates, sometimes they have 10 isolates. So of course, it's not really comparable. But at the same time, you can see that there are differences between phyla and different classes of the microorganisms. So therefore, it's kind of against the hypothesis that microbes are distributed everywhere. So it's just, we see, at least on the planktonic microbes in different oceans, we have a different composition of these microbes. So what are the take-home messages? Again, bacteria and archaea are two dominant groups, especially if we're talking about deep sea waters, and diatoms will be present on photosynthetic biofilms. Next generation sequencing is a good tool to investigate biofouling and biofilm composition, and we understand and know the depth, orientation, and the type of substratum. It means that protected, unprotected substratum will affect biofilm's composition. What are the challenges of investigation of biofilms in deep sea? So as you remember, maybe the device that we use in the Mediterranean, where you have panels, one above each other, there was a release mechanism that allowed us to retrieve it up to the surface. And then, of course, it can be contaminated by the microbes that live in the photosynthetic area. So this might be one of the major challenges. So how to protect and prevent these deep sea microbial communities from contamination during the retrieval of your samples. There are also technical difficulties with the deployment, replication, because we need to see the replication For example, with the glider studies, we deployed 2 gliders, one was lost, and then we end up with the limited number of replications. It's required the special equipment. It's also costly because you need to have a vessel. You need to also if you're doing molecular analysis, so you need to pay a lot for molecular analysis. Development of the communities takes time, especially if we are talking about deep sea, where the temperatures are around 0 or even slightly below 0 or +5 maximum. So it will take months. So you need to think about the experiments that will not be conducted in 12 days as it was conducted in Australia, but in maybe 1 year or so. And it is difficult to investigate the role of the microbes, how they interact in these complex microbial biofilms, what kind of compounds they produce, and so on. And at the end, if you are using molecular techniques, then you might also end up having false results with the ghost DNA. So it's DNA that exists outside of the living cells and just trapped in the biofilm. So what are the future directions? We need definitely to look more into the deep sea biofilms. In particular, we need to look at the certain groups like archaea that are under-investigated, viruses, and protista. We also need to do a deep sea experimentation. So not only look at the one type of substrata, but different types of substrata, different time, different depth, different locations. We also need to employ new tools, such as, for example, autonomous vehicles or different omics approaches. And we need to understand the role of these microorganisms in the deep sea biofilms. At the end, I would like to say thank you. And of course, thanks for the funding agency. So this is a picture of the glider that we were deploying. The investigation was done together with my colleagues from United Kingdom, and also, of course, funding agencies from Oman. Thank you very much.
Thank you very much, Sergey. That was an impressive presentation and very interesting, and I have lots of questions, but I think we will initiate the discussion later after all the presentations. And now I would like to invite Dr. Kerstin Meyer-Kaiser from Woods Hole Oceanographic Institution to tell us about recent advances in deep sea biofouling.
Thank you very much. I am excited to present today. I am going to discuss 2 different studies that I have been involved with recently. One is in the Arctic deep sea, examining biofouling, and the other is developing a new technology. So I'll begin with our long-term experiment. This was conducted in the Hausgarten area in the Fram Strait with my collaborators at the Alfred Wegener Institute. It was initiated in 1999 by Michael Fages from ABI. I was not a part of the study at that time. I was still in primary school. But I was invited to join the study when I was collaborating with ABI starting 10 years ago. So the lander was deployed in 1999 and visited with a remotely operated vehicle in 2003, and then again in 2011. That was my first expedition with ABI. And then in 2017, we decided to recover the metal frame, which you see here in situ on the seafloor. And I was invited to do the analysis of the macrofouling on the panels. So I'll show you some of the ROV footage that we have. In 2003, you can see that there are no animals visible on the panels. There were a few cut off from the experiment at that time and examined in the laboratory. There were foraminiferans present, but no metazoans. In 2011, you can see that there were some hydroids visible on the panels from the ROV. Those kind of scratchy lines on that panel are the stolons of a hydroid. In 2017, there was actually not much visible either. There are white serpulid tubes, and again, the hydroids were visible. But the macrofouling really became apparent when I examined the panels under a dissecting microscope on board Polarstern. these are the organisms that I found, and these top 4 are foraminiferans. I have a very difficult time identifying foraminiferans, but I was able to get this panel C to a species that is Cibicidites wollersdorfi, which is very common suspension feeding benthic foram in the Arctic. E, F, and G are all sponges. E, I believe, is a hexactinellid sponge, possibly Calophacus arcticus. F is Tinea abrissorum, which is very common in the Hausgarten area. And G is the carnivorous sponge, Cladorhiza gelida. It was very exciting to have a carnivorous sponge on our panels. We also have a hydroid and a soft coral here. This is Garsemia. As you can see, an anemone, a stardulid, and a crinoid. This panel L was very interesting for me. I first assumed that it was the stalked crinoid, Bachycrinus carpenteri, which is very common in the Hausgarten. But on further examination, I realized that it was actually a pentacrinoid. This is the early life history stage of a feather star. And so it was very cool for me to see that we have an early life history stage that then swims away and engages in a pelagic life as a feather star. So one of the advantages of this experiment is that we are able to examine not just what is on the panels, but how those macrofouling communities relate to some environmental factors. So the bottom current is very, very consistent in the Eastern Fram Strait. It is to the northwest, usually between 7 and 8 centimeters per second. And so I knew which panel was located where on this metal frame that we had for the experiment. And so I was able to compare the communities of fouling fauna that we had on the panels based on their substratum, because we had brick and plastic, based on their height off the bottom, because we had panels deployed on the top and the bottom of the frame and also on the inside, and also based on their angle of orientation to the predominant bottom current. And as you can see here, there were significant differences in the macrofouling on panels with different construction. So the plastic had generally lower abundance than the brick panels. In fact, on the back side of the brick panels, there were triangular ridges. And in those depressions, there were high abundances of individuals, especially of that hexactinellid sponge. I assume that it was taking advantage of the microhabitats provided by those ridges. We also have a significant difference in the fouling on panels at different locations on the metal frame, on the top, on the inside, or on the bottom. And there were higher abundances of many suspension feeding taxa on the top panels, which I assume is because they have an advantage being elevated in the benthic boundary layer for suspension feeding. And there was no significant difference based on panels of different orientations to the bottom current, except for one species, for the hexactinellid sponge. It had 3 times the abundance on panels that were in line with the predominant bottom current, which again I attribute to suspension feeding, because those hexactinellids are inflexible. They're not able to change their morphology, such as the anemones and the soft corals and the crinoids can do, in order to accommodate themselves to the bottom current. So it was very interesting to conduct this study and see the Arctic fouling over 18 years. We can draw some conclusions based on this study. First of all, the recruitment was very low. We only had 13 species compared to the regional species pool, which is somewhere between 40 and 60 morphotypes of hard bottom fauna. So it was a very small portion of the regional species pool that recruited to our panels. We can conclude that the Arctic deep sea hard bottom community takes decades to develop. Our experiment was deployed for 2 decades. And again, we had that very low recruitment, and the community on the panels did not match what is present in the background community. And I also believe that the recruitment may be stochastic and patchy, especially because we had so many young microscopic individuals of some of these species, suggesting that they had recruited to the panels right before its recovery, potentially in stochastic or patchy recruitment events. This experiment provided a lot of insights into recruitments in the Arctic deep sea. The second study that I would like to talk about today is one that I've been working on with collaborators at Woods Hole Oceanographic Institution. I wanted to design a camera system that could be used to monitor biofouling in remote environments. And so I approached some of the engineers that we have on our staff. You'll see their names here to the right. Yogi, Stefano, and John have been extremely helpful in this study. Karis Schrag is my PhD student who is also involved with this. And so we call this the camera to analyze invertebrates, or contain for short. And this is a camera system that is contained within a housing, but we use the end cap of the housing as a fouling panel. So the basic design of the camera system is here. You will see that we are using the clear end cap of the housing as the fouling panel itself. So it's clear, and I use that as the settlement surface. We have embedded some lights around the edge to provide illumination. On the inside, there's a machine vision camera. We wanted to get very high resolution, so we chose a 24 megapixel camera with a macro lens. It is positioned a set focal length from the panel, so it's always getting those images in very good focus. We have a small computer, a Raspberry Pi computer, as well as a Sleepy Pi computer. These are just basically little circuit boards, and the Sleepy Pi wakes up the Raspberry Pi, which wakes up the camera, so that the battery is only turning on when it is time to take an image. And so we can deploy the camera for long periods without running out the battery. And the settlers are viewed from the underside of that panel. I'll show you some example images. We have conducted a few test deployments, unfortunately not in a deep sea environment yet. This is only under the pier in Woods Hole, Massachusetts at 16 meters. But still, we can use these test deployments to gain some important information. This is one example image. You'll see that the scratch marks are where I have roughened the clear end cap with sandpaper to provide some rugosity to increase the settlement. You can see the illumination from the sides. That's from those lights that we have wrapped around the end cap. And this is a snail, Asteris lunata, which is crawling on the panel. And so you can see that there is the shell a little bit out of focus because it's not flat on the end cap of the panel. You can see the foot, which is in focus on that end cap. And I believe this might be the radula of the snail, which is potentially grazing as it crawls across our camera system. Another example image, this is from a month-long deployment from earlier this summer. And at first it looks just like a whole bunch of dots, but the images are high enough resolution that when you zoom in, you can tell what is there. So we know that this is a barnacle, Balanus, which is a very common species in Massachusetts. I believe this is an ascidian, or a tunicate, Botuloides violaceus, which is a very common fouling species. And what's really neat about Botuloides in this case is that we can use the camera system to tell when it is dying. Look at that post-settlement mortality, which is usually very difficult to observe. So we see the living individual here on the top, and a few days later, it is degenerating and dying. So we can measure post-settlement mortality with this. And this is one more example image from later in that month-long deployment. You can see it's very populated with macrofauna. Obviously, it's a shallow environment, so you get heavy settlement. And again, we see that same barnacle, which has now grown. So we are able to measure growth rates of very young benthic species with this. This is an encrusting bryozoan, which is here. You can see the ancestrala and then watch the colony grow over time. This is another bryozoan, Bugula tirida, which has a tree-shaped form. So the yellow halo is the majority of that colony, which is out of focus, but you can see where it is attached on the camera. And we also have a polychaete, tube-building worm that has settled on this system. So currently, Catane is a working prototype. We have built one, and it is working at 16 meters. So my PhD student, Karis, is currently working on the image analysis pipeline using Python in order to build up a method for analyzing these images by drawing polygons around each of the settlers so that we can track their growth rate and their mortality over time. Our next step is to make this camera system more user-friendly and available to other researchers. I would love to make this broadly available for usage. And my vision is that eventually this can be used to measure settlement rates over time, post-settlement mortality, growth rates of organisms right after they have settled. It could potentially be used to monitor overgrowth competition, for example, in sessile colonial species that may overgrow one another, track animal movements such as that snail, and it could potentially be modified for experiments. For example, if a certain species is made to settle on the camera in the lab and then it's deployed in different environments in the field. I would love for this to be useful for Arctic and Antarctic environments, could be deployed on deep sea moorings, remote locations, or anywhere that a researcher cannot access on a daily basis. So we are currently working with the engineers to try to make this system user-friendly and able to be produced so that it can be purchased and used by other researchers. With that, I thank you for your attention, and I am happy to answer a few questions.
Thank you very much, Kirsten. It would be an underestimation to say that I'm amused by the camera. It's just really cool. And okay, I would like to process to other presentation, which is mine. And I would like to tell you about the use of autonomous seafloor equipment for studying biofouling below the photic zone. Well, in the last decades, we can observe the rapid expansion in deep sea by various industries and mainly oil and gas, which leads to increase of number of artificial constructions on the seafloor below the shallow water zone. And these objects provide the hard substrate, which is literally precious on the soft bottom. At the same time, fouling communities worldwide have mostly been studied within the first 50 meters of the ocean. While it comes to the deeper areas, there is definitely a massive knowledge gap. So it can be partially explained by technical and financial difficulties, especially in the deep sea, lower than 300 meters. And existing methods turn to be really labor consuming when applied to these areas. And of course, we can still rely on different observational methods, such as regular sampling or video shooting by remotely operated vehicles. But experimental methods allow us to control different aspects of the biofouling process, including type and state of the substrate, exposure time, and location of the study. So that's where the test plates come to the scene. They are made of glass, metal, or plastic, or they can be with or without different coatings. They are commonly used as artificial substrate to monitor the succession of fouling communities at various depths, and especially in the shallow waters. They are simple and cheap. They can be installed for various duration. They are easy to install or to deinstall, to preserve, to transport. They are convenient for analyzing the qualitative and quantitative characteristics of the community. And the question is, how do we deliver the plates to deeper regions? One can use separate special frames just for this purpose, but we believe that there is another cheaper opportunity for this. We can use seafloor equipment as monitoring platforms. The plates can be mounted onto the different rigid elements of this equipment for the duration of the study, which can, in our point of view, really broaden the range of depths and geographical locations, as well as reduce costs of biofouling research. And actually, any autonomous seafloor equipment that remains stationary for some time has the potential to accommodate these plates. In support of this method described, I would like to present the case study from the Sea of Okhotsk. It's a marginal sea of the Western Pacific Ocean. In the mid-July 2019, we mounted some steel and plexiglass plates. It's shown on the photo to the buoy frames of autonomous buoy stations. And there were 2 study sites that were located approximately 50 kilometers eastward from the Sakhalin island. Each station had 2 acoustic Doppler current profilers and their buoys with metal frames, which were used as the platforms for the plates. There were 8 plates in total, 1 plexiglass and 1 raw steel plate attached to each of the 4 buoys. And each plate was A4 format. So there is a detailed scheme of the ABS on the slide. We had panels here and here. And after a 3-month deployment in mid-October, the stations were collected and the plates were detached and then analyzed in the laboratory. What did we see? Well, steel plates were covered with rust, actually. We presume that it is the second bacterial stage of the biofilm succession in the shallow waters. So the bacterial community modified the surfaces of the plates into more complex landscape, and we presume that it became more suitable for settlement of larvae of eukaryotic fouling species. We also analyzed the samples of the rust using just light microscopy and observed something that we think are bacterial colonies. We also saw something which looks like crystals, but of unknown origin to us, and we're not sure if they were formed after the preservation or before. So we found no traces of multicellular organisms. What we would really like to achieve in the long run is the identification of the members of this bacterial assemblage. We realize that it requires more complex analysis, genetical analysis, as well as sampling for scanning electron microscopy or probably something else. For now, it really exceeds the abilities of our laboratory, so we're open to any collaborations in this field. And plexiglass plates. Well, 2 plexiglass plates which were mounted at 45 meters were absolutely clean, and they showed no visible signs of micro and macro fouling, but probably they had some invisible biofilm, which we didn't really test. Another 2 plates submerged at greater depths at 150 and 230 meters showed 2 different stages of colonization with more complex biofouling organisms. We observed the traces of complex microfoulings and microfouling and macrofouling. The plate from ABS2 at 150 meters was densely covered with various sessile ciliata, different sessile infusorians. So we had 2 species of ciliates from subclass Suctoria. It is Ephelota gemipara and Acineta compressa, and 1 colonial species of the subclass Paratrichia, which we could not really identify due to its poor condition. There was also a small specimen of hydroid. There is no photo here because it was severely damaged and we could not really identify it beyond the order level. Another plate from 230 meters contained 1 juvenile barnacle, which is presumably Hirona evermanni, which is a common species for this region, and few individuals of severely damaged Ephelota on the one side, while the other side was free of any visible macro or microfouling. I believe that we could have done more to analyze the biota from these plates if only we had some more proper experience of working with these tiny creatures, because we're all like macrozoologists and this was a wholly new world to us. And now after, for example, after struggling to look at these gigantic samples, A4 samples, I think we should use smaller plates for periods less than half a year at similar depths in this region. The emptiness of the plate is a bit puzzling to me, and I'd really like to discuss this later with the colleagues who know more about microfouling and its developmental patterns. So although these results, they do not provide enough data for proper analysis, there is definitely some strong evidence of biofouling both for steel and plexiglass plates. So we believe that this approach have proven effective for future biofouling studies, and the test plates were deployed and collected and have attracted some fouling organisms, which is a positive result. But there is a lot to be modified and a lot of to be tried, of course. For example, we would like to work in some new locations. Now there are some plates in the Kara Sea and in the Laptev Sea. I think we will, some of them are already collected, and we will collect some more next year. We would like to work in other seas in the Arctic and in the Far East region. Also, different duration of the panels exposition at various depths, preferably below the photic zone, is at stake. Actually, we would like to, well, to test the plates for several years or more than for several months. We would like to try new materials and probably to collaborate with some other interested parties. And also, we are going to make the panels less dependent on the rigid frames, because sometimes the equipment does not allow installment of the plates to the frame. Some seismic equipment, for example, is, you cannot really install anything to the frame. So we would like to make the panels, less dependent on this. Thank you for your attention. And I'm looking forward to some discussions after the presentations. Our next panelist is Petra from St. Abbs Marine Station. And Petra, we are welcoming you.
Thank you. Hi, everyone. My name is Petra Harshaini. I'm a supervisor and researcher in St. Abbs Marine Station in Scotland. And today I would like to share some of our research on understanding the effects of electromagnetic field originating from offshore renewable energy devices, how they affect microorganisms. And in this talk, I would like to talk about how they could affect biofouling. Scottish Renewable Energy currently has 950 megawatt operational and 4.1 gigawatt consented offshore wind farm project. The newest one, as you can see on the map, the orange area, the North Nagoya, gonna occupy 105 square kilometer area. So with this recent increase in large-scale renewable energy developments in Scotland, extensive areas of seabed at risk of exposure to elevated electromagnetic field levels emitted from the cables. Industry standard cables can be effectively insulated to prevent electric field leakage, but they are unable to prevent magnetic field emissions. And as with any anthropogenic emissions, it is crucial that we understand the levels and the extent of the emissions. Uncertainties concerning the impacts of EMFs on marine species, particularly marine organisms, are very important that are addressed. In the context of renewable energy and biofouling, any submerged surface in the marine environment goes through the process of biofouling with the formation of bacterial biofilms and macromolecular film, and followed by macrofoulers. But unfortunately, it's a very costly industrial problem, shortening the life expectancy of these devices, and the removal and disposal of this increased biomass proposes another problem for an industry. The other aspect of biofilms and renewable energy side is usually these sites are fisheries exclusion zones, but it is expected that the subsea power cables and scour protections laid on bare substrate could serve as artificial reefs and could potentially provide refuge for commercially important species and potentially contribute to stock replenishment. And as initial colonizers, bacterial biofilm have a crucial role in facilitating the settlement of some of these macroinvertebrates as well. With regards to electromagnetic fields and the effect on bacteria, there is already scientific evidence that certain electromagnetic field, depending on the source and the intensity, could affect the cell mobility, growth, morphology, and even antibiotic resistance. In this research, to assess the effects of electromagnetic emissions on early biofouling on subsea power cables, marine biofilms were subjected to static electromagnetic field at St. Abbs Marine Station. We are situated at the east coast of Scotland with the Berkshire Marine Reserve on our doorstep. It is a purpose-built facility with very minimal magnetic and electromagnetic footprint, therefore it's an ideal place to conduct magnetobiology research. For this study, to mimic the EMFs expected around high voltage direct current subsea power cables, we used Helmholtz coils and solenoid magnet to create magnetic flux densities between 0.5 to 10 millitesla intensity levels. And the microbial biofilms were developed on fiberglass mesh tiles and constant raw seawater flow. And to compare the effects of different EMF field intensities, biomass was evaluated using crystal violet assay. And as the extracellular matrix has a crucial role in cell adhesion, to assess the total carbohydrate content of the matrix, we used the phenol sulfuric acid method. And to determine changes in metabolic rate due to acute EMF exposure, a two-dimensional oxygen imaging system, the VisiSense imaging system, was used in this study. What we found is EMF had an effect on biomass and the extracellular matrix as well. 1.5 and 3 millitesla EMF exposure resulted in decreased biomass, and also what we found that it weakened the extracellular polymeric matrix as well. When we looked at the oxygen depletion rate of the tiles, so it's the blue area that's increasing on the videos, we found that the same field intensity level had actually decreased the depletion rate of these tiles as well. Our results presented here showed that static EMFs at strengths expected around subsea power cables impact early biofilm formation in a field intensity dependent manner. And to further understand the underlying mechanism of electromagnetic field effects on early biofilm formation, the collected DNA samples will be analyzed to see whether EMF had affected the bacterial community structure and diversity. And we would like to adapt this methodology and repeat it in a field experiment as well. But based on our findings, it is clear that in order to aid future EMF research, there is a need for accurate field strength measurements from around the power cables. And to fully understand the impacts of EMF from renewable energy development on marine species, it is crucial that we assess the effects at different magnetic field intensity levels. Thank you.
Thank you, Petra. That was really interesting. So our next panelist is Dr. Sergey Alenin from Klaipeda University, and he would like to tell us about biofouling of marine infrastructures. Please, Sergey.
Yes, thank you for the invitation, and I really was excited to listen to this very innovative and highly theoretical presentations. My talk will concern practical aspects of biofouling mostly, and you know, this is a huge field, and I will mostly speak about decommissioned marine infrastructures. Okay. Well, this is a snapshot of marine traffic with different types of ships and roads, and all of these ships, they have ballast water, which is transported all around the world, and that's why it is called ecological roulette. And some of these species which are being transported and released in different coastal ecosystems, they may become invasive. It means what they may establish, multiply, spread, and cause problems to human health, economy, and environment. And this is a well-recognized problem. And to solve this problem, there is a IMO Ballast Water Management Convention, which is actually a legally binding document, which is now in force. And this is another it's subset of the previous snapshot, which shows the special craft, including platforms and tug vessels, which have much less ballast water, but much more biofouling. And there are no IMO guidelines, only not legally binding recommendation. But it was recently estimated what approximately 60% of all established coastal and estuary and non-indigenous species are transported by biofouling. When we compare biofouling on ships and oil platforms or rigs, we can find at least 10 differences. And well, starting from the depth of immersion, wetted area, which in oil platforms is much deeper, or usually platforms may have complex 3D structures. Then ships are, of course, mostly in transit, while oil platforms, when established, they are stationary. And probably most important thing is what every ship, after some period of time, they go through the docking and they are being cleaned, while oil platforms, they have either not at all any docking period, or usually biofouling is not removed when the platform is being towed. And for ships, and the collisions of alien species usually take place in ports, while oil platforms, they may act as a hub, as a stepping stone from service vessels. So this is one of the examples of the docking area in Spain, in Gran Canaria. This is a docking area for rigs in Las Palmas. This is a really big business for local economy, very important business. And rigs are being tagged to this hub, and then from there they are moving to different places, to Mediterranean Sea, to Arab countries, and so on, the Far East. So in recent investigation, they found 11 species of non-native species, fishes in this very area. And this means that not only sessile biofouling organisms can be found, also associated fish as well. This is another example about the about oil platform, which was towed from Brazil to Singapore, and after 55 days in the middle of the ocean, there was storm conditions, and they released this platform. And after 1 month, it was found Tristan da Cunha. And analysis of biofouling show that there are species which may be harmful for local aquaculture, for local lobster which is there. Today it was told, we were heard already about the biofouling stages, and usually macrofouling appears after several weeks. Please correct me if I'm wrong, but this is what I found in literature. And it is after that time, after several weeks, the biofouling starts to work. I mean, at the special infrastructure start to work as a stepping stone. Stepping stone appears. To best of my knowledge, there is no biosecurity risk assessment of such a platforms. The only example is Australia and New Zealand. They recently established anti-fouling and in-water cleaning guidelines, which require to assess the risk. Domestic biofouling means no risk, or regional or international biofouling is high risk, and they also have a very detailed and concrete proposals how to manage the risk of biofouling. To avoid the stepping stone effect, first of all, this just a few words about Rigs to Reef. This is a program. You know that there are a lot of decommissioned infrastructure, right? And people think what to do: to sink it into the deep ocean, to take it out for metal, or to use them for good, to enhance habitat diversity, increase habitat diversity. So the recommendation would be these new habitats, they shouldn't be placed in the areas of ship ballast water operations, where discharge or uptake of ballast water is taking place. Also close to thermal discharge from power plants in our coastal industry, in ports and marinas, close to ports and marinas, in ship repairing yards, and in aquaculture installation. Well, in conclusion is what the decommissioned marine infrastructures act as stepping stones for marine invasive species. It was proving. Biofouling management of ships and oil rigs is really different. So there should be different approaches here. And a specialized legislation, rules or code of practice is needed for management of oil platforms biofouling. And biosecurity risks should be taken into account in the rigs to risk program. Thank you.
Thank you very much, Sergey. Now we have, well, a bit less time than planned for the questions and discussion. If anyone has any questions, you are welcome to ask them. There is a question for Kirsten. At what depth the camera was deployed?
Yes, the first test deployments are just under the pier in Woods Hole, Massachusetts. The depth is 16 meters. It's quite shallow.
May I? Just a question for our colleagues specialized in biofilm. Do you have any idea of experiment or setup of the study to understand the role of biofilm for next stages of succession? From which side we can touch this question, if possible?
Okay, so I understand that the question is primarily to me, so I will try to answer. It depends on what kind of environment that you are talking about. So if, for example, we are talking about Arctic or Antarctic, I absolutely agree with Kirsten. She showed the data, and we also done similar experiments in Antarctic, and it takes months, years even, to develop macrofauning communities. So even if you prepare such experiment, it will be difficult to conduct and difficult to monitor. But if you're thinking about the shallow water communities, like maybe 10, 20 meters depth, then it's, I mean, boreal communities or some sort of tropical communities, then in this case, the simple experiments, you again have the settlement panels, and then you develop a biofilm. You either develop a biofilm in different conditions and then move them into this environment, and then look at the formation of macrofauning communities. Recently, we've done an interesting study that actually will be interesting to another Sergey. It's about invasive species. So it was done with my colleagues in Portugal. And we developed biofilms in different areas. So there were areas with the anthropogenic impact. There were areas without anthropogenic impact. And it was areas with the protected areas, simply. And then we shuffle them around. So we move from one area into another area. And then we look at the macrofauning communities formation. And we see that actually, if we move a biofilm from a protected area into the area which is anthropogenically impacted, we have less invasive species formed. So in fact, it is important not only to monitor invasive species or to have a nice environment, but also you need to have some sort of special biofilm that will be an indicator for those species that this is a good environment to settle and to live.
Thank you very much, Sergey. We have another question, from another Sergey.
This is from Sergey to Sergey. What was the difference between? Did they measure, your Portuguese colleagues? Did they measure the level of pollution in in this affected anthropogenically affected areas and in marine protected areas? Was it a real difference between this, or how it was, or just geographically they choose this and this?
No, there were difference, of course, geographically, but it was a difference also with regarding of pollution as well. Yeah, and surprisingly for me that these biofilms actually maintain different within several months. So one can do such an experiment, maybe develop a biofilm in certain conditions and then move it into another conditions and then monitor what kind of macrofouling species will be formed. Even after several months, you will see the differences. But again, regarding deep sea, I cannot tell you because deep sea succession process is rather lengthy, and you might need to wait years to see the differences.
Okay, thank you, Sergey. And we have a question for Kirsten. So is your PhD student, I assume working with Python, is looking into artificial intelligence for image analysis?
Not my PhD student, but one of my collaborators, Yogi Girdhart, is actually a specialist in machine learning and artificial intelligence. And so this is the plan, that he will use the images we are collecting during the test deployment to try and develop automatic methods for image analysis.
Okay, thank you very much. So there is also a question for Petra. Are you or your colleagues looking into EMF and settlement of macrofouling organisms?
Yes, that would be the next step. We have a lot of research on invertebrate, like commercially important species, crabs and lobsters, and how it affects their physiology and behavior. And as their early life stages is very important and it's less known, we would like to do settlement trials with these species as well. So definitely in the future, we will look into that.
Thank you very much, Petra. And there is also a question about our steel panels. So do we know the grade of the badly corroded steel panels? That is a good question. Actually, no, we don't. But we hope to collaborate with people who do, who actually are specialists in corrosion, and I expect some new results from these collaborations. Thank you. I also have a question for Sergey on biofilms. Are there any ideas of how you could isolate the panels from the surrounding environment while taking them out? Is this problem is somehow being addressed? Because it's really a problem.
Regarding, I think the question was regarding the contamination with the microbes of surrounding waters. So yes, you can develop certain devices that actually will enclose the panel before after the deployment and taking into the surface, and then it will be incorporated in the surrounding waters. What we did with my colleagues from Greece, so we just simply, since biofilms attach firmly to the surface, we assume that whatever is attaching during the retrieval of these panels will be attached loosely, and then we just wash this. So whatever is washed, it means that maybe these are those microbes that didn't attach firmly, and biofilms usually attach firmly, and then you just analyze those ones. But yes, if you want to be precise, you would like to make some sort of housing that will be closed, and then once you take this panel, you will take it with the water surrounding it, and it will be not affected by other microbes.
Thank you, Sergey. And I have another question for Kirsten. How did the biofouling in the Arctic experiment compare to surrounding communities?
Yeah, we actually have some pretty good understanding of what is in the surrounding communities, thanks to over 30 years or 20 years of research in the Hausgarten area. So the species richness was very low compared to hard bottom communities in the area. We were missing several of the abundant species that are common on the hard bottom reefs and on dropstones. So it was, it was very low species richness and very low abundance compared to what was surrounding.
And there is a question about the depth in the Arctic experiment. What was it?
Oh, it was 2,500 meters.
Okay, thank you. And another question. So is the lack of records of viruses and biofilms due to lack of virologists studying them, lack of suitable technique, or lack of viruses? And could ghost DNA be of viral origin? Sergey, I suppose that is to you.
Yeah, it's an interesting question. I don't know the clear answer. Probably it's due to many different reasons. First of all, how you can study viruses in the biofilms. So basically, you need to sequence using a shotgun metagenomic sequences to get the sequences of all microbes and then identify and separate the viruses, viral sequences out of them. And for this, of course, it's required the money. The second probable reasons that we don't know much. Maybe viruses are playing an important role in biofilms and therefore in biofouling. We simply have no answer for that. And if we knew that they are playing an important role, then probably will be more studies focusing on that.
Thank you, Sergey. So I think that we're actually running out of time a bit, and I would like to invite Professor Vadim Makiyevsky to say some words in conclusion of this impressive session.
Already, I just start to learn. I expect it to be continue more and to learn much more about the session, because there are questions I still did not got an answer. Is the question about the role of biofilm and modification of biofilm for the next stage of succession. You described well how it can develop, but we do not know what is the differences on these stages and how fast and how in which extent it can affect the next stages, and especially on the stage of settling of macroorganisms. The second, I guess the data, I did not know, for my shame, I did not know about this experiment with biofouling in house garden. However, I am in since the beginning of this long-term observatory, but I miss this experiments with biofouling. And it seems that this data, the time scale of the development of fouling on 2000 meter fit well to what we know for the depth of 200 meters. The staging is again one order of magnitude longer. And the question is, do the stages elongate proportionally, or the stages, and we have longer stages on the first or the beginning of fouling succession, and then it appeared to rotate faster and faster. Because with Sasha, we saw we got unexpected results, very long duration of this initial phase for 3 months, which drive us to the stage of, let's say, 2 weeks in shallow Baltic Sea. But at the same time, 6 years old community on artificial constructions looks typical for every hard substratum. It looks like kind of terminate stages of succession. So it looks like that it speed up on the next stages. For that, I hope the installation is still there in house garden. It's a pity because we need more time. So that is 2 things we got from this, at least 2 important things, that there are approaches, there are it's kind of real things to investigate biofouling, and I hope we will find more dense contacts to collaborate in this field and to send you our plates or whatever. And it seems that altogether we come to create the general time scale of deep sea artificial or hard substratum community development. It's what we proclaim in recently written manuscript, hopefully will be published, where we proclaim to create altogether the general timetable of cold water succession, let's say, because we have just very few points of very different duration. That is most important 2 outcomes. And the next, I expect a kind of communication and coordination using addresses. I don't know, Anya, how we will manage with that, how to keep the community of about 30 people who are interested in the matter, how to continue the communication. Thank you. A lot of thanks for teachers.
Well, I think that there are still too many questions and a lot to be discussed. And personally, I would like to discuss more. And probably we will in due course organize some kind of seminar or something like that to discuss some some problems that we had no time to discuss now. And I would really like to thank all of you for very interesting presentations. They're very different and all of them are really, well, they're excellent. Thank you very much. And I hope to see you sometime soon. Thank you.
Thank you very much, Alexandra. And with that, unfortunately, we're bringing this session to the end. Thanks again for all the speakers. Thank you, Alexandra Khairidinova, for being our technical support during this meeting. And thanks for all the participants to your questions. We'll be in contact and hopefully there will be more discussion coming after the session as well. Thank you and goodbye.